So this is a chain of how protein is produced inside a cell. Most of you know these things. So we have the DNA inside the nucleus. Then DNA then gets transcribed into a messenger RNA. The messenger RNA swims its way into the cytoplasm through the nuclear pores. Then once in the cytoplasm, the ribosomes come into the messenger RNA, they assemble themselves and start producing protein. This process is called translation. The protein that is getting produced is an amino acid chain of the protein. This is the primary structure of the protein, and it's a premature protein. To get fully mature, it has to get folded into the 3D structure of the functional protein. The folding occurs by itself, and now we have a 3D structure produced by the ribosomes. Now, the bound ribosomes give that protein into the rough endoplasmic reticulum. The protein passes through the membrane and goes inside the rough endoplasmic reticulum. Once there, the rough endoplasmic reticulum adds some carbohydrates in a process called protein glycosylation in order to increase the stability of the 3D structure. Those proteins and the glycoproteins then get sent into the Golgi apparatus via a transport vesicle. The Golgi apparatus is considered as the packaging plant of the cell, مصنع التعبئة والتغليف. So what it does, it labels these newly produced proteins with molecules that are specific to the destination site. So if a protein is designed to go and work as an enzyme in a lysosome, it gets labeled with a molecule specific to the lysosome. If this protein is part of the cell membrane, it gets labeled with a molecule that specifies that it is going to the cell membrane. If this protein is a hormone to get secreted outside the cell, then another molecule gets labeled and tagged to this protein. And this changes the newly produced proteins into fully mature proteins. These fully matured proteins are ready to go to their destination sites. So the Golgi apparatus put them in vesicles, for example, secretory vesicles that secretes proteins outside the cell, or transport vesicles. Okay, these vesicles move around in the cell via a motor protein. This is not required from you, but it's fascinating to know that there are some molecules, proteins, that function and moves with legs that walk on the microtubule. This is a type of cytoskeleton, and carrying the vesicles by their arms. This is really legit, and this is true visualization of the kinesin, a type of a motor protein. Anyway, continuing, so the ribosomes are two types. We have the free ribosomes and we have the bound ribosomes. So the free ribosomes are the ribosomes that are floating in the cytoplasm. They produce proteins that function inside the cytoplasm, for example, the enzymes. While the bound ribosomes, they are bound to the rough endoplasmic reticulum, and they produce proteins that are secreted outside the cell, that goes into the cell membrane, or that goes into the lysosome and other organelles. Okay, so the ribosomes are the site of the protein synthesis. They are two types: the free ribosomes, located freely in the cytoplasm. They produce proteins that function in the cytoplasm, for example, the enzymes or the cytoskeleton. And we have the bound ribosomes. These are located on the surface of the rough endoplasmic reticulum. They produce proteins that function in cell membrane, lysosomes, or to secrete outside the cell, for example, the hormones. Now, the eukaryotic ribosomes are big compared to the prokaryotic ribosomes. They have an 80s in density. This is a unit in density, and each ribosome has two subunits: the small subunit and the large subunits. The small subunit has a density of 40s, and the large subunit has a density of 60s. When these combine and form one ribosome, these don't add up to 100 but add up to 80s. And each subunit from these contains ribosomal proteins and ribosomal RNAs. Okay, now the endoplasmic reticulum. We have two types of the endoplasmic reticulum: the rough endoplasmic reticulum and the smooth endoplasmic reticulum. They are rough because they have granules of ribosomes attached to them, while the smooth don't. The rough endoplasmic reticulum functions in protein glycosylation, secretion of proteins and glycoproteins to Golgi apparatus via transport vesicles. The smooth endoplasmic reticulum function in lipid synthesis, the phospholipids and the cholesterol, carbohydrate degradation, metabolism, for example, the glycogenolysis, and lastly, functions in detoxification of drugs and toxins, poisons, for example, the alcohol and the barbiturates. Okay, now this is the structure of the Golgi apparatus. It's made of multiple cisternae. These are membranous structures. We have the cis face, the receiving side, and the trans face, the shipping side. Now, what it does, as we said, it transforms the newly synthesized proteins into mature proteins by tagging and labeling each protein with specific molecules specific for their destination sites. They do that by removing sugar monomers from the glycoproteins, producing diverse oligosaccharides. So this is what it does. Golgi apparatus, other names Golgi body and Golgi complex. So it transforms newly synthesized proteins into mature ones. They tag proteins with molecules assigned for their final destination, like removing sugar monomers, producing diverse oligosaccharides. This is the sorting cell products function, and they store and package proteins in vesicles, ready to send them to their final destination. This is considered the packaging plant of the cell, مصنع التعبئة والتغليف. So moving on to lysosomes and peroxisomes. Both of these are membrane-bound organelles. They are vesicles, originate in the Golgi apparatus, and contain different enzymes. So the lysosomes contain hydrolytic enzymes like the lysozyme that hydrolyze any organic material, carbohydrates, lipids, proteins, and nucleic acids in acidic environment. They are the digestive compartment of the cells and have three functions. They get rid of cellular waste, the older proteins. So the proteins with time, they might change their structure and become dysfunctional. So the cell recycle them, degrade them into amino acids for the production of new proteins. This destruction of old proteins is called autophagy, الابتلاع الذاتي. Okay, so the second function is to destroy the cell when the cell is damaged. For example, when the cell gets infected with viruses, the lysosomes try to destroy the cell by a process called autolysis, التحلل الذاتي. The third function is to destroy pathogens and harmful materials after phagocytosis. Okay, so we have this bacterium and this cell is a white blood cell. The bacterium gets attached to specific receptors on the white blood cell, then it gets engulfed, phagocytosed, and the phagocytic vacuole is produced. This phagocytic vacuole combines itself with late endosomes, other cellular waste in endosomes, and lastly, the vesicles from the Golgi apparatus carrying lysosomal enzymes combine with both of these and form a mature lysosome. The lysosome then break down the bacteria. So now the peroxisomes, on the other hand, contain catalase enzyme, and this catalase enzyme is very important because it speeds up the breakdown of hydrogen peroxide H2O2 into water and oxygen. This hydrogen peroxide is toxic to the cell, and the peroxisomes detoxify it. Peroxisomes are plenty in liver cells. Now the vacuoles. So the plant cells, for example, have central vacuoles. All vacuoles are surrounded by a membrane called tonoplast, and the central vacuole in a plant cell store food and water. Other types of vacuoles include the phagocytic vacuole we just talked about, this one. It isolates harmful and waste material. And lastly, we have the contractile vacuole that regulates cell osmolarity.